All right, we are on chapter 10, section 3, and we're going to talk about the three-dimensional structure of proteins, because proteins don't exist as just a string of amino acids, right? That's all we've talked about so far. And that's really just the very basics. That's the primary structure of a protein. So it's the order in which our amino acids are joined. So if you look at it and you look at your side chains, right, you can decide which amino acid you are looking at. And so that's you look at the protein backbone, and then you look at the side chains, right? The protein backbone is just a repetitive sequence of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, right? That's the backbone. So there are things that are attached to the backbone, like you have all those hydrogens that are attached to the backbone, and then you have the carbon with the double bonded oxygen, right? And so that's attached to the backbone. But then, right, you have the side chains. Those side chains are basically dangling from the backbone, and they're hanging out in water, and lots of them don't like to be hanging out in water. So what happens is they end up folding into three-dimensional shapes, which then give the protein some function, right? So just how they're lined up from N-terminus to C-terminus is our primary structure. Our secondary structure, it occurs when you have the protein that is going to either coil into a helix, kind of like a really old telephone that has a cord that coils, like that's in my office. So I guess not too old. Or you can have repeating patterns of what are called beta sheets. So you can have alpha helices, which are the most common, that kind of look like the telephone cord. And then you can have beta pleated sheets, where it's like a sheet that goes this way, and then it kind of makes another sheet here, and then it makes another sheet there, and then it makes another sheet here, and they're kind of connected. And so it kind of goes up and down and up and down and up and down. And so the way that these secondary structures are beneficial and are held together is by interactions with the protein backbone. So with those hydrogens that are dangling down here, so all of these hydrogens and these oxygens, right? So you end up having the hydrogens from, let's say, one strand of a beta sheet interacting with the oxygens on another strand of the beta sheet, right? And so that's what stabilizes these secondary structures. And so remember that when this happens, these aren't covalent bonds, these are hydrogen bonds. We have hydrogen interacting with that oxygen, or remember, no fun, nitrogen, oxygen, or fluorine. In this case, it's always going to be the oxygen. But remember, it's not covalent. It's just an intermolecular force. So it's very strong for an intermolecular force, but it's still just an intermolecular force. So here are some examples of the secondary structures. This is the alpha helix. And so in this alpha helix, we have hydrogen bonds between the carbonyl of our oxygen atoms. So remember that carbon double bonded to the oxygen, and the nitrogen that has that hydrogen is going to interact with the oxygen. So remember that the hydrogen is partially positive, which is why we do that little sigma sign. And this oxygen is partially negative right there. And so these two things are attracted to one another in a hydrogen bond. And remember, we used to do those hydrogen bonds as little dots. And so that's how we indicate a hydrogen bond. And in an alpha helix, it's very, very particular. It's that the amino acid 4 amino acids away is what actually ends up interacting. So if you counted, right, here's 1, and then you would have 2, 3, 4, and 4 would be making that interaction. And it just so happens that that's how the coil works, that you have that stabilization every about 4 amino acids away, right? And so it's really important because it allows the helix to kind of stretch and come back and stretch and come back, right? And so it's not in a perfectly rigid shape. And that's really how molecules work. They're not actually solid in the way that we think of things being solid. They're constantly moving. You know, like I think of them like my 9-year-old. They're constantly moving, always changing, always doing something, right? And so having a lot of these hydrogen bond interactions helps to keep this a very, very strong structure. And so when we look at those amino acids, that's the backbone interactions. But what's going on with those side chains, right? Because we have all of these side chains here in blue. What are they doing? Well, all of those side chains in a alpha helix are pointing outward. So in this model, all of the R groups are purple. So here you have an R group, here you have an R group, here's another R group, here's another one. This one's kind of pointing in the back, right? All of these are really just sticking out. Do you notice that? They're all sticking outwards, away from the axis of the helix. And so they can end up interacting with other alpha helices, or maybe they're going to interact with a beta sheet. But they don't participate in secondary structure. Secondary structure is only backbone interactions. Only backbone interactions. So you're going to have to differentiate between secondary and tertiary structure, which is the next one. So secondary is only backbone interactions. So here's another secondary structure. This is the beta sheet that I was talking about. So here's like the beginning of it, and then the backbone kind of comes up this way, and then it curves around, and here's another sheet. And then it curves around, and here's another sheet, right? And so you end up having these side by side. This is 3, right? 1, 2, and then you get a third over here. And those are held side to side together by hydrogen bonding. So it's again hydrogen bonding between peptide backbones. So here's our carbon double bonded to an oxygen. Here's our nitrogen bonded to a hydrogen. And this is where we put those dots in and we say, okay, that's the hydrogen bond. And so this zigzag structure allows it to swap. So the first hydrogen bond is the carbonyl on one side and the amide on the other. So you go down to the next one, and the next one's going to see, oh, I have a nitrogen bound to a hydrogen. Here's my carbon double bonded to an oxygen. So here's my double bond, my hydrogen bond, right? So they're held together again by interactions in the backbone. So on the beta pleated sheet, what happens with those side chains is the side chains are either projecting, if this is the backbone itself, they're either projecting out like into the screen towards you or behind towards me, right? But the side chains are not interacting in that secondary structure. They are, they're hanging out and they're waiting for the next interaction. So how important is protein secondary structure? Well, it turns out super, super important. And one case where we can see this is Alzheimer's disease. We know Alzheimer's disease is a form of dementia, right? And people have memory loss. They have very foggy thinking, right? And they have trouble performing normal daily tasks. And it's progressive. It gets much, much worse. And typically it only affects adults who are over the age of about 65. Not always, but mostly. And if you look at the brain tissue of people with Alzheimer's, what you're going to notice, there's some very different morphologies, right? So brain tissue in people who have Alzheimer's have these plaques, that are between the brain's nerve cells. And so it's a combination of plaque buildup, and not plaque like your teeth, not that kind of plaque, but I'll show you what plaque we're talking about in a second. And then inside the cells, the neurons themselves, you have neurofibrillary tangles that occur inside the neuron. And so it's a combination of plaques outside of the cell and these tangles inside of the cell that interfere with nerve impulse transmission, right? And then when the nerve cells can't talk, they end up dying. And so the brain tissue actually shrinks. And so if you look at a brain of an Alzheimer's patient and the brain of a normal patient, you'll see that the Alzheimer's patient brain has basically shrunken because it's lost so much mass, right? So what are these plaques? Well, these plaques are actually proteins. They're fragments of 42 amino acids long, and they're called the beta amyloid protein. And so if you look at beta amyloid protein in normal patients versus Alzheimer's patients, what you're going to see is they're structurally very different. It's the same 42 amino acids, but how they arrange is different. So if you're a normal person, your beta amyloid is alpha helical, like my telephone cord. But if you have a person who has Alzheimer's and you look at their beta amyloid protein, what you'll see are beta strands. And so that change, the primary amino acid sequence is the same change in secondary sequence from that alpha helice to the beta sheets is what forms the plaques. Because those beta sheets are basically sticky, they are attracted to one another instead of being kind of not attracted to one another. They are now because of that secondary structure, and they form those plaques. And so we don't know how to fix it. There's no cure for Alzheimer's disease. You can slow the progression with drugs. You can lessen the symptoms for a limited amount of time by regulating the brain's neurotransmitters, but we don't have a cure for it. So that's an ongoing thing. So if you look, this is the difference in protein secondary structure, right? This is somebody who's normal. They have alpha helices, whereas over here we have beta sheets. And so the amino acid sequence is the same, but in the alpha helices, they don't get very close to one another. But when we turn it into beta sheets, then they stack up one on top of the other and they form these plaques here. And so these beta amyloid plaques are outside of the cell, but you got to think that cells have to communicate with one another. So if you have this big plaque sitting in between the two cells that are supposed to communicate, they're not really going to communicate. And there are some neurofibrillary tangles that occur inside the cell as well. But that's it's a different thing. Okay, so we did primary structure, that's amino acids. Secondary structure, that's interaction of the backbone to make things like alpha helices and beta sheets. And now we have tertiary structure. Tertiary structure is where our alpha helices will interact with other alpha helices, beta sheets will interact with other beta sheets, or alpha helices will interact with beta sheets, right? But it's these secondary structures interacting with one another in order to create the tertiary structure. So why does this happen? Well, for a couple of different reasons, right? So if you have nonpolar side chains, these are hydrophobic, right? They are repelled by an aqueous environment, right? So if they're sitting on the outside, they're like, oh, I want to get away from the water, and they kind of fold inward. And so now you have other residues that are exposed to the water, and these hydrophobic ones are poking on the inside of the protein and are sequestered away from the water. And so they're happier that way, right? Whereas you would have polar side chains here that would hang out and be like, oh, hey, water, let's make some hydrogen bonds and that kind of stuff. And so you'll see them on the surface of proteins that are normally exposed to an aqueous environment. And so those tertiary structures end up being stabilized by attractive forces between side chains and attractive forces between the side chains and the environment. So the polar ones, right? So these would be polar amino acids with water, right? And then attractive forces between the side chains themselves. So the side chains themselves may interact and make an intermolecular force. And we'll talk about what some of those examples are. But this is side chain to side chain. Ooh, if I could write today. So this is side chain to side chain. So all of those interactions help this protein to fold into a specific three-dimensional shape. It's not just, okay, here's an alpha helice, here's a beta sheet, but maybe it's alpha helice, beta sheet, alpha helice, beta sheet, right? Okay, so your book has some cool drawings of, here's one protein, right? And this one polypeptide has primary sequence, right? Because it has a particular amino acid sequence. Now it also has alpha helices, right? So this looks like an alpha helice to me. Okay, and then it also has, here's a beta sheet and here's a beta sheet. And so what you can see are that you have hydrogen bonds between the beta sheets, right? Between the backbone of the beta sheets. So that'll give you a secondary structure. You also have hydrogen bonds between the backbones inside of your alpha helice, even though they didn't draw them in, but they are there, right? So that'll give you secondary structure. Now, what's going to give you tertiary structure? Well, sometimes you'll have, here's one alpha helix and here's one alpha helix. So if you have a positively charged side chain and a negatively charged side chain, you have a positive and a negative. They're attracted to one another. So normally when we have a positive and a negative ion, we call that a salt, right? Like sodium chloride. But in this case, because it's inside of a protein, and not just an ion, we call it a salt bridge. But it's just a salt bridge is an ionic interaction, right, between side chains. You can have hydrogen bonding. We already talked about hydrogen bonding. You can have what are called disulfide bonds. So if you have two groups that have SH, right, and then you have SH, and you have the rest of the amino acid backbone there, right? So what happens is these two hydrogens will get pulled out. So we'll erase those. And what you end up forming is a bond between the two sulfurs. So we call that a disulfide bond. So I can link an alpha helice and a beta sheet together with that, right? Let's see. You can have hydrophobic interactions. So here I have two side chains that hate the water. So what ends up happening is that they fold, so now they can interact with one another through London dispersion forces instead of trying to hang out with water that's polar that it doesn't really like, right? And then the last one is that you can have actual interactions between, here's a side chain and here's a water molecule. And so you can have hydrogen bonding occurring between the amino acid side chain and the water. That helps to stabilize. So all these things help to stabilize getting to the tertiary structure. Okay, so tertiary, what kind of tertiary structures can you have? Nonpolar interactions. That's what we showed. A nonpolar wants to hang out with a nonpolar because they're repelled by the aqueous environment. And remember, aqueous environment is just saying water, so that they can be on the inside of the molecule. So nonpolar interactions are London dispersion forces, right? Because that's all nonpolar molecules can do. Then you can have polar interactions where your polar amino acid side chains are interacting with water through dipole-dipole. Well, it's really hydrogen bonding, right? And so you could say, oh, they did have it. Okay, so it could be dipole-dipole, ion dipole, or hydrogen bonding. They just did them in a different order, but it's cool. So any of those would be polar interactions. Then you can have salt bridges. Salt bridges are specifically ionic. So you have to have a positive and you have to have a negative interaction. And so usually it's between a basic amino acid and an acidic amino acid. And then they're attracted to one another, and that's the salt bridge. And then you can have disulfide bonds. So just like we said, you have two SH groups, which are thiols. They react, and it's an oxidation reaction, right? Because you're losing hydrogens, and then you form the disulfide. And you can add different chemicals to make that happen. All right, so here's kind of an example of that disulfide bond forming, right? We have our amino acids on our protein backbone. And so here's our two thiols, and those two thiols are going to undergo an oxidation. So if we go this way, we are oxidizing, which means that we are removing the hydrogens. When we remove the hydrogens, both of those sulfurs need another bond. They need more electrons to complete their octet. So they will form a bond together. So this is really kind of cool because this is how perms work, right? So what you do is you chemically treat somebody's hair in order to reduce the disulfide bonds. And so now the collagen molecules in the hair can kind of move and orient, and you put them in rollers, right? And then you attach those hydrogens, so you undergo a reduction, and then you lock them in place while the hair is in the curlers, and then chemically, right, you've changed the shape of the hair, and when you take the curlers out, the curl stays. So that's a perm. It's kind of cool. Okay, So tertiary structure, like we said, is how all of those beta sheets and alpha helices interact with one another. And so what ends up happening is you get the overall shape of a protein from this three-dimensional structure. So globular proteins form basically a spherical shape, right? And you have those polar amino acids that hang out on the outside, and you have the non-polar forming the core because they're pointing inwards and they don't want to interact with water. And lots of enzymes and some different cellular proteins are globular proteins. You can also have fibrous proteins that are, they look more like rope, right? They're thread-like. And so what ends up happening is a lot of times you have, like in hair where you have keratin, you have one alpha helice coiled on another alpha helice. And those alpha helices are linked together with those disulfide bonds. And so that's what happens when a perm, right? If you have straight hair like mine, it's stuck in this orientation. But if we reduce those bonds, now the ropes can basically slide against one another. We curl up the hair on the curler, and then we remake those disulfide bonds. Now it's stuck in the curled shape, right? Same things in collagen. So in our skin looks nice and beautiful and healthy because we have lots of collagen, but as we get older, we don't make as much collagen. And so that's why we end up having wrinkles and things like that. All right, and they tend to be, right, your hair, your skin, they're very durable. They're very tough structures. All right, so we talked about primary is the sequence, secondary is the interaction of the backbone, tertiary is the interaction of side chains to create an overall three-dimensional shape. Now we have to talk about quaternary structure. Quaternary structure is when you have multiple peptides that have to come together to make a single biologically active protein. Sometimes you don't need a quaternary structure. This is like optional. Some need multiple polypeptide chains in order to function. Some proteins won't function unless they have 4 polypeptide chains or 8 polypeptide chains, and some only need 1. So the individual polypeptide chains, or sometimes we call them subunits, are held together by the same interactions that stabilize tertiary structure. So side chain interactions, disulfide bonds, all those kinds of things, right? And not all biologically active proteins need a quaternary structure. Some of them only have tertiary. So this is kind of just a reminder of the different forces that stabilize each of the levels of structure. So here's kind of just a visual image of the different ones. So A is primary structure, right? Our sequence of amino acids. B is whether we're going to make a alpha helice or a beta sheet. Tertiary is when we have multiple alpha helices interacting with maybe a beta sheet and other things, right? And it gives you an overall 3D shape. So this is a globular protein, right? And then, oh, this one needed 4 subunits in order to be functional. And so this 4 subunit protein has a quaternary structure because it needed more than 1 subunit. So let's go answer some problems. All right, so what kind of bonding is present in our primary structure of a protein? So primary structure, you got to remember, primary structure is just our sequence of amino acids. So our sequence of amino acids are held together only by covalent bonds, right? These are our peptide bonds. All right, how many different, that's for class, name the stabilizing attractive force found in secondary structures of proteins. Now, be careful in reading, right? Stabilizing attractive force. Attractive force tells me it's not a bond, but we call it a bond, which is so terrible. So it's hydrogen bonds, right? Because in secondary structure, this is making things like alpha helices, beta sheets, but all of these are backbone interactions. And the only thing that you can interact with in the backbone, right, is that you have that carbonyl carbon, and then you have the nitrogen, right, and it's bound to the rest of the amino acid, and that's bound to the rest of the amino acid. But then there's a hydrogen, and so then you can make that hydrogen bond between those two. Okay. All right, let's see. Describe the differences in the shape of an alpha helix and a beta pleated sheet. I just end up calling it a beta sheet. All right, so our alpha helix. So here's our alpha helix. Let's talk about that first. So in an alpha helix, it's coiled, right? So you remember you kind of snakes, if you kind of snake it around, right? That's a terrible, terrible one, but that's okay. And you make those hydrogen bonds along the backbone. So this is coiled, right? It's amino acid side chains. They point outward. Remember, so these amino acid side chains kind of like just stick outward. They don't interact within the one helix, right? Point outward from helix, right? So there's coiled amino acid side chains, point outward. What else? Oh, I drew it, but I didn't say it. You have hydrogen bonding along the axis of the helix every about 4 amino acid residues, right? Okay, so then a beta sheet. So in a beta sheet, remember our beta sheet kind of looked like this, and then you had it where the points would come in, and then it was hydrogen bonding between those points, right? So it's the same thing in that our hydrogen bonding occurs between the backbones, between the strands of the backbones. Hydrogen bonding, did we say that? Hydrogen bonding along the axis of the helix every 4 amino acids, but in both, we'll say here, both, the hydrogen bonding occurs in the backbone, right, in the backbone. So this beta sheet, so this is in both, this one and this one. But beta sheet specifically, right, this is kind of a zigzag. We drew it this way. So this is a like an open, extended zigzag structure. I don't think I've ever written that before, of beta sheets, right? So your amino acid side chains are either going to orient. Remember, we said you have that zigzag, and then they're going to extend above or below the sheet. So side chains extend above and below sheets. Okay, and I think that gives us our differences between the two. All right. What type of interaction would you expect between the side chains of each of the following pairs of amino acids in the tertiary structure? Okay, I think we kind of need to name what are the possible types of interactions. So we'll kind of do this along the side. I guess we have room down here. We'll do it down here. So here we're going to do types of interactions, right? So then we know what we're choosing from because they don't really list it. So one, if we have nonpolar, if we have nonpolar residues, then these would be London forces, London dispersion forces, right? This would be the hydrophobic effect, because they're trying to get away from the water. They're all nonpolar, so they want away from it. So they want to fold inside, and they're going to interact with each other with London forces. Then you could have polar, right? That's the opposite of nonpolar. So if you have polar interactions, gosh, that could be anything, right? That could be hydrogen bonding. That could be dipole. Kind of depends on what your structure is, dipole, or you could have ion dipole interactions, right? But that's things that are hydrophilic, right? 3, you could have salt bridges. Remember, salt bridges are always going to be where you have a positive and a negative amino acid side chain. Let's see. So you would have something that's basic, that's positively charged, and something acidic that's negatively charged, right? 4 would be a disulfide bond. This is actually a bond, right? This is not an intermolecular force. And this would be, you would have to have 2 thiols, THIO thiols, which is that, right? Something bonded to a sulfur, bonded to a hydrogen, and they form, you reduce them and you lose those hydrogens, right? And form the disulfide. Okay, so those are our types of interactions. So now we have to kind of look at our pairs and see what we know about them. So what do we know about lysine and glutamate? So go look at your table if you have to. But lysine's positive, right? It's got that, it's got a bunch of CH2 groups. It's got 4 of them, and then bonded, it has an NH3, and that nitrogen's positive. That's just the side chain. That's just the R group. I'm not drawing the whole amino acid. And then glutamate has only 2 of them, and then it has a carbon, and that carbon's double bonded to an oxygen and bonded to another oxygen. So this would be a positive and a negative, right? So that would be a salt bridge. Let's see, leucine. Leucine is nonpolar. Isoleucine, it's just a different configuration of the carbons, right? So this is also nonpolar. So the most that could happen between 2 nonpolars are London forces. Threonine, threonine has an alcohol, has an hydroxyl group. Okay, so we're gonna put that. Tyrosine, another alcohol. So if we have 2 alcohols, what can they do? They can hydrogen bond. So you can have a hydrogen bond there. Glutamine has an amide group, and that amide group is polar. It's not charged, but it's polar. And then arginine has a protonated amine. So if we have a protonated amine, that's positive. So a positive and a polar would give us an ion dipole interaction, right? So you just kind of have to get to know your amino acids. Have fun with them. All right, 10.29, determine whether each of the following statements describes primary, secondary, tertiary, or quaternary structure of our protein. 3 polypeptide chains interact to form a biologically active protein. So we have 3 polypeptide chains coming together. That's telling me it's quaternary. If you have to have more than 1 polypeptide chain to be active, then that is quaternary. Hydrogen bonds form between adjacent segments of the backbone, of the same protein to form a folded fan structure. That's a beta sheet. It's a beta sheet. Okay, so because it's between the backbone, that's telling me it has to be secondary. Nonpolar side chains are repelled by water to move to the interior of the protein. So now we're talking not between the backbone, now we're talking between the side chains. So the side chain's telling me it's quaternary. Amino acids react in a condensation reaction to form a peptide bond. It's primary. It's just primary. Okay, let's see. 10.31. We're going to do 10.31 and 10.32. So what's the difference between the secondary structure present in the beta amyloid protein in a person without Alzheimer's and in that with Alzheimer's? So if you do not have it, so if you are without your beta amyloid, right, protein folds one way. Folds into what? It folds into an alpha helix, right? Or alpha helices, E-S-E-I-X-E-S, helices. Okay, anyway, but if you have Alzheimer's, you have the same protein, it's a beta amyloid protein, but it folds in this case into a different shape, which is the beta sheets. So how do the beta amyloid plaque form, right? Well, the beta amyloid protein folds into beta sheets, Now the beta sheets aggregate, and that aggregation, right? So they stick together. They stick together, form plaques. And these plaques are between, I was going to write my abbreviation BTW, between neurons. And then the neurons can't communicate. NEUR. Neurons can't communicate. And the neurons die. And that's why we have all those symptoms. All right. Hope you have a great rest of your day. Yep, that's the end. All right. See you next time.